All right, let's get right into it. If you really want to get a handle on lymphomas, you can't just start by looking at the cancer itself. Nope, you have to go all the way back to the beginning, to the development of the B cell. And you might be asking, why? Well, it's because the entire modern system of diagnosing lymphoma is built on understanding this exact developmental journey. It's like a biological story that gives us the map we need to find our way through these incredibly complex diseases.
You know, it almost always starts this simply. A patient walks into the clinic with a swollen lymph node. That's it. No huge dramatic story, just a node. And this one single simple finding, it's the first clue that kicks off a really deep and complex investigation.
So the first move is usually an excisional biopsy. Under the microscope, the pathologist might see that the normal structure of the node is just gone. It's been completely overrun by a sea of similar-looking lymphocytes. That's a massive red flag for lymphoma, for sure. But it is not a diagnosis. It's kind of like showing up to a crime scene and knowing a crime was committed, but you have no idea who did it or how or why. Just saying it's lymphoma is diagnostically, well, it's useless.
And this gets to the absolute heart of the matter. Just looking at the cells, their shape, their size, their morphology, it's just not going to get us to a specific answer. The central, the absolutely indispensable tool in our toolkit is immunophenotyping. What we do is we use this whole panel of protein markers on the cell surface, we call them CD markers, to create a unique fingerprint for each lymphoma. And that fingerprint tells us its precise identity and where it came from.
Okay, so here's our game plan. We'll start with that diagnostic challenge, then we're going to trace the B cell's entire life, from its birth in the bone marrow, through its journey as a naive cell, its big activation moment in the lymph node, and then its final destinies. And we'll wrap it all up with a practical matrix that shows you how to put all this amazing knowledge to use.
Our story kicks off deep inside the bone marrow. This is where a common lymphoid progenitor cell makes a life-altering decision. It commits to becoming a B cell. And that decision is marked by expressing a key protein on its surface, CD19. I want you to think of CD19 as the family surname that a B cell is going to carry for its entire life. It's the ultimate B cell marker.
So this development in the marrow unfolds in three really distinct stages. First up, we have the pro-B cell. It's got that family name, CD19, along with some immaturity markers, CD34 and HLA-DR. Then it matures into a pre-B cell. Now this is where it gets really interesting. It sheds those immaturity markers and picks up two critical new ones, CD10 and an enzyme called terminal deoxynucleotide transferase, or TDT for short. TDT is a really specialized DNA polymerase that's a hallmark of immature lymphocytes. At this stage, it also finishes rearranging its immunoglobulin heavy chain gene. Finally, it becomes an immature B cell. Here, it drops the CD10 and TDT, and now that both the heavy and light chains are sorted, it can proudly display its very first antibody on its surface, IgM.
And this developmental pathway gives us a perfect example of the link between how a cell develops and how it becomes cancerous. When these precursor cells in the marrow turn malignant, the cancer you get is precursor B-cell acute lymphoblastic leukemia, or ALL. The most common type actually comes from that pre-B cell stage, which means it's CD10 positive. And in kids especially, having this CD10 marker is actually associated with a good prognosis.
So our B cell has finished its basic training in the bone marrow and gets released into the bloodstream. At this point, it's known as a naive B cell, and it's starting a whole new chapter of its life out in the periphery.
We call this cell naive for a simple reason. It hasn't met its specific antigen yet, so its whole life is basically a waiting game. And if it doesn't get activated, its default biological program is to undergo programmed cell death, apoptosis. The vast majority of them, well, they're just destined to die.
But what happens when that self-destruct program fails? When these naive B cells just refuse to die and instead start to multiply? Well, the result is chronic lymphocytic leukemia, or CLL. So CLL is fundamentally a disease of the peripheral blood. It's a cancer of failed apoptosis. And as part of this malignant change, the CLL cells pick up two new, very characteristic markers, CD5 and CD23.
Okay, so for the lucky few naive B cells that get rescued from that fate of apoptosis, their journey takes them to a really important place, a lymph node. This is the activation zone. You can think of it like a university campus, where B cells go to finally meet their antigen and complete their education to become super effective defenders of the body.
Now this lymph node campus has distinct zones. The B cell first arrives in the pre-germinal center area, which we call the mantle zone. From there, it moves into the heart of the action, the germinal center, which is buzzing with cells called centroblasts and centrocytes. And finally, after its training, it exits into the post-germinal center or the marginal zone.
Now, sometimes B cells can turn cancerous in that very first zone, the mantle zone, and that gives us mantle cell lymphoma. And here's where things can get a little tricky, because it shares a marker with CLL. Both are CD5 positive. So how do we tell them apart? The key differentiators are right here on this slide. Mantle cell is CD23 negative, while CLL is positive. Even more, mantle cell is positive for surface IgD and a protein called cyclin D1, which is a key driver of cell proliferation. Both of those are negative in CLL. This right here is immunophenotyping in action, solving a real clinical puzzle.
So inside that germinal center, B cells are going through this intense process of mutation and selection to improve their antibodies. It's a high-stakes environment, and cancers that arise here reflect their specific cell of origin. Those rapidly dividing centroblasts can give rise to really aggressive cancers like diffuse large B-cell lymphoma and Burkitt's lymphoma. The more mature centrocytes, on the other hand, can lead to follicular lymphoma. But even though they're different, they all share a common germinal center signature. They're all positive for CD10, CD23, and a master regulator protein called BCL6.
After graduating with honors from the germinal center, our B cell is now at a crossroads. It has two potential career paths it can take, two final forms it can become, to help provide long-term immunity for the body.
Okay, so path number 1 takes it through the marginal zone, where it becomes a long-lived memory B cell, just waiting, ready to pounce if the same infection ever comes back. Path number 2 involves a pretty dramatic transformation into a plasmablast and then, ultimately, a plasma cell, which is basically a tiny, powerful antibody factory. And as you might have guessed, each of these career paths has its own set of malignancies. If things go wrong on the memory B cell path, you can get marginal zone lymphoma, small lymphocytic lymphoma, which is really just the lymph node version of CLL, and hairy cell leukemia. On the other hand, on the plasma cell path, a malignant plasmablast can cause a very aggressive subtype of DLBCL, while a fully malignant plasma cell is the cause of multiple myeloma.
So, wow, we've just traveled through the entire life cycle of a B cell. And now we can take all of that developmental knowledge and boil it down into a powerful, practical diagnostic tool that pathologists use every single day to solve these cases.
This simple matrix is just beautiful. It perfectly summarizes our diagnostic logic. Just by looking at the pattern of 3 key markers, CD5, CD10, and CD23, we can slot a lymphoma into one of 4 major buckets. Okay, let's walk through it. If it's CD5 positive and CD23 positive, you're thinking CLL or SLL. If it's CD5 positive but CD23 negative, you should be suspecting mantle cell. If it's CD10 positive but CD5 negative, that points you straight to a germinal center lymphoma like follicular. And if it's negative for all 3, what we call triple negative, your top diagnosis becomes marginal zone lymphoma. It's such an elegant system, and it's rooted completely in the cell's own life story.
So we've seen how this developmental map lets us make these incredibly precise diagnoses. The final question, the big one, is what's next? Knowing exactly where a cancer cell came from, what its story is, and what biological machinery it relies on to survive, that does more than just give the disease a name. It gives us a target. So how can we leverage this incredibly deep understanding to design smarter, more effective therapies that attack the specific Achilles heel of each lymphoma subtype? That right there is the challenge and the incredible promise for the future.